Hi, everybody. I've been given the thumbs up to get started. So let me start by welcoming everybody to the 2020 LabKey User Conference. Absolutely our first virtual user conference. So we're looking forward to a new experience with all of you and hope we're going to make it as interactive and dynamic as we can. So first off, I got to say thank you. And a little different this year, I'm going to say thank you first to my colleagues, the employees of LabKey. It's been a challenging year for everybody, and the LabKey employees have just been fantastic. They've had individual challenges as well, and they've worked through it, and they've continued to support all of you, and it's been incredible. And I'm so appreciative of the opportunity to work with these folks and to be part of this team. I also want to thank all of you. The majority of you on this call today in this meeting are researchers or support the research process. And this year, more than any year, has reminded us just how important that is. The work you do to help everybody, to help humanity, to help the world is incredible. And you inspire all of us. You're the reason why people come to work at LabKey and continue to work at LabKey. And I don't know if you hear it enough, but thank you. We appreciate it. I want to thank all the speakers this week. They took a lot of time, both the internal and external speakers, preparing and being able to share their experience with all of you. So I want to say thank you to all of those folks, and thanks to Will for helping to coordinate our unique and first virtual user conference. So by way of history, I don't know how many of you are familiar with history, but we started at the Fred Hutchinson Cancer Research Center, now the Fred Hutch. And we started because the Macintosh lab was dealing with an explosion of data, having too much data, and Excel and other systems weren't enough to support that data. And they needed a platform such as LabKey. So the founders of LabKey went to work for the Hutch and created this platform. And this platform has grown and scaled throughout the years, and the number of use cases, the number of different labs, the different geographies that we've supported has also grown. And the company has grown from becoming a standalone company through the years and working with some fabulous partners. And you'll hear that word often from us. Those partnerships are so important to the growth. And to our starting having roots at a nonprofit research center makes you a different company. And it's part of our DNA, part of who we are. More recently, as we've shifted to also building products, built for purpose products, as you've seen in the last few years. And then a couple of really key milestones for us in this year. First off, earlier this year, we launched Sample Manager. And you're going to hear the product manager, you're going to hear Hannah speak about that on Thursday and give some demos and show you how Sample Manager works. And then most recently, we've been working on freezer management, where we heard we also need from our users to have stronger freezer management capabilities and solutions. We launched a few years ago LabKey Biologics, but this year, our users, in particular, our friends at Just said, we really need an integrated electronic lab notebooks. We've been working on that this year. And you'll see that presentation by Bernie, by the product manager, on Wednesday. You'll also hear presentations by folks at Just as well. The other big thing that happened this year was the work we've done with the FDA to launch the COVID My Studies app, which is built around getting e-consents to help with the consent process for trials, COVID-related trials during this time. So we'll share that presentation on Friday, and you'll get to see more about that. And then finally today, we'll get to hear from the presenters at NIAID about the work we've been doing there to help with the studies they've been doing to combat COVID-19. So there's a lot of work that you're hearing from us and from our partners to support COVID research. This is just a few of the ones that we've been working with, but of course we wanted to highlight some of the groups and the research they're doing to get us back to normal. So thank you. As I said, right from the beginning, we've been built with researchers for researchers. We weren't some folks who just wanted to create a software company that we saw a market for. We were called to action from researchers and said, we need your help. We need your partnership. And that approach is a unique approach. It's something that's very important to us and to how we work. This comes out more in person, I think, when we get to spend a few days with you at a user conference, but hopefully the folks who've been working with LabKey will vouch for this. And this unique approach is started by the community edition of LabKey Server. Right from the beginning, having a free platform for researchers and available to researchers has been so important to us. So we continue to support this enterprise platform and make it available for all researchers because we believe in the cause. The other thing that's important about LabKey is the flexibility. We know there's commonality across labs and there's solutions that can be leveraged and reused. and scaled. But we also know that the way you work is unique, and there are some differences among how you practice your science, and we have to have that flexibility. So all of our solutions have that flexibility in mind to be able to tailor it for your needs. So what I'd say is this week, as we're doing our presentations, if there's something that you see and it doesn't quite meet the need, but you think it's close, please ask us. Take the time post the user conference to inquire, can this be changed? Can this get adapted? Because the answer is almost always yes. The other thing that's unique about us is the incremental, that faster time to value. That comes with both our development methodology in terms of agility, but that also shows up in our products, where we believe you shouldn't have to wait for years to see the value of working with a software vendor. You should be able to see it quick, because that helps you get the adoption in your lab. We hope that helps you get others to want to use the software, and that makes for a richer solution. So having faster time to value has been so important to us. And then finally, the partnerships, the ability to work with you, to support you. Like I said from the beginning, is what inspires us. And those partnerships have been going on since the start of the company, and we really enjoy them and relish them. So thank you. So the solutions of LabKey are candidly countless, because we have a platform that is so robust and that can do so much for supporting informatics. The use cases are also countless. We highlight a few here just to kind of call out some of the bigger ones and some of the areas that we've been supporting. But those solutions really are diverse and varied. And they're supported by, at the heart, LabKey Server, really underpinning everything we do. But our three core products are LabKey Server, Sample Manager, and Biologics. And you'll hear about each of these products all 4 days this week. Other thing I think that's become very apparent to everybody this year is it's important to work with a company that has products and services that meet your need, but also for many of us, it's important to work for a company that has a core set of values that match your values. We realize you have choices. And so what we wanted to share a little bit in the morning is, what are our values? What can you expect in terms of how you work with LabKey? What do LabKey employees get to expect from each other and from me? So our values, some of these are the same, but one of the things that we felt this year in particular, we really wanted to add was a value around diversity and inclusion. That matches and goes with our values in terms of innovation and flexibility, accountability, collaboration, passion, yes, drama, no, and the support for the community. As I said, this year was a very unique year. And one of the things it caused us to do is to really assess, how are we doing? And the answer is we were doing good, but not good enough in terms of our commitment to diversity, equity, and inclusion. So many of our employees, almost all of the employees, worked together for several weeks to say, how are we going to put a statement that matches our values, that's aspirational, that matches where we've been, but where we're going. And what you can see here is our commitment to DEI. And at the heart of that is the strong belief that true innovation comes from a mix of diverse ideas, diverse experiences, and diverse people. And with that, that's how we're going to propel the company continue to grow. And LabKey is committed to diversity, equity, inclusion at all levels of the organization. I also realize it's one thing to have a statement, it's another thing to put it in action. So I wanted to call out some of the commitments that we have historically made and that we're going to make to DEI. And this list is going to be a list that grows over time as we learn from all of you, as we learn from each other, as we learn from the community. But this at the heart are some of the commitments that we've made to DEI, okay? In terms of compensation, in terms of our workplace flexibility, in terms of how we promote, in terms of our objectives and how we measure our success, in terms of how we give each other feedback, in terms of how we can do better. Our other commitment remains to the community. This year was a little different, so Girls Who Code, which is one of the groups that we really enjoy working with, and it was really meaningful. We weren't able to do some of the hosting on site, so there were other types of commitments that were made to that organization. You can see the photo from past years. We have an internship program because we think that's a way for us to give back and to train the next generation. We participate in the science fair. We donated money to the food banks. And finally, as a company, our commitment to our employees is everybody gets 7 days a year, which they can use either for their own education or for volunteering in their community at the charities of their choice as a way to give back. So a few conference highlights. Hopefully you've already seen the agenda and email or on the website. But really at the heart, the purpose of the conference is for you to have a chance to learn from LabKey users and from our team. Now it's a little different. We can't do all the breakouts that we like to do in person, but we still think we have a great week of content and presentations. And I'm really excited to get the feedback from all of you as you see LabKey sample managers, you see biologics, as you hear about the plans for server, as you hear from other users, to let us know what you found interesting, what helped you learn, and what we can do better. So please give us that feedback. We will be recording the sessions. We realize not everybody can commit to 4 days. We tried to do it 4 days to make it a little more imagine. But we will be recording the sessions and sharing those recordings. So first off, today, after me, I'll hand the presentation to Adam. He's one of the founders of LabKey. He's also responsible for product management, and he'll walk through an update for the LabKey server. Look back at the year and look forward. We're going to have presentations today from both NIAID, from Sandhya and Jason, and also from Nestlé, from Bernhard. And then we'll cap the day with Rob, head of program management, talking about reporting and visualizations via ODBC. And there'll be time for questions throughout the presentations. Day 2, heavy focus on biologics, the work we've been doing in ELN, presentation from Tara, and then anchoring the day with Josh giving a presentation on Panorama and the work we do for targeted mass spectrometry. Finally, day 3, Hannah and team will be presenting LabKey Sample Manager and the work that we're doing on freezer management, presentations from Patrick from OHSU, and then a presentation from Steve in terms of showing how what you've seen throughout, but especially on that day in terms of Sample Manager, how you can extend that further in a LabKey server. Finally, the 4th day, which we usually call Tech Talks, there'll be a handful of different presentations from the LabKey technical team in different areas that we've heard from all of you would be interesting. So please join with us for as many of those days as you can. Give us the feedback along the way. Thank you again for the partnership and for the opportunity to work together and support what you do. Next up is Adam, who'll be talking about a LabKey server, the product update there.
So, very excited to see all the attendees, all of you who could join us for this conference. We, like Michael, very excited about the conference, very excited to see all of you folks, and looking forward to 4 days of great content. So I am going to focus on LabKey server. Many of you know a lot about LabKey server, but just as an intro, LabKey server was our first product, and for a long time our only product. And still today, all of our other products, whether it's Sample Manager or Biologics or Panorama, My Studies, they're all built on top of LabKey server as a platform. So we think it's important for you to understand what we've been working on for the last 12 months or so in the way of LabKey server and making LabKey server better, enhancing it, making it more reliable, full-featured, because that is the foundation that we build everything on top of. Now, I'm going to be talking about the last 12 months and talking about what we're doing for the next 12 months. I've got about 14 minutes to do that, so it's kind of rapid fire. I've tried to focus on just the most important key things. I encourage you to look at the release notes for every release of LabKey, to look at the documentation, and of course, as always, talk to your account managers and other LabKey folks to get more details about these features and the hundreds of other features and enhancements that we add every single year. So I'm going to start first with what I sort of label infrastructure improvements. So these are features, they're maybe not the most visible features, but they are very important, critical to LabKey working with in modern day environments and in your infrastructure, important for security hardening and for administrators and developers. So that's sort of the key message here is we've done many, many improvements to LabKey over the last 12 months to help administrators, to make their jobs easier, and to help developers, both the LabKey team, where many of us are developers here, to make our lives easier, but also for our clients who also do a large amount of development on top of LabKey. We want to make everyone more efficient, make it easier to develop and enhance LabKey. So first of all, one of our favorite features, we completed a very large project to completely redesign and rewrite all of the field editors in LabKey. So the field editors are the components that allow administrators to customize all the different table types in LabKey. So data sets, lists, sample types, many other different types of tables are customized using field editors. And we made a big change. We completely redid the user interface, completely rewrote it. This has a couple of benefits. One is it's just much easier to use. It's more intuitive, easier, more full-featured for administrators. It also allows us, as the developers of LabKey, to add new enhancements and new capabilities very quickly and easily. So very excited about that. That development also leveraged a lot of the user interface paradigms and technologies that we developed for Sample Manager and for biologics. So you'll hear me talking about this integration of these products with the LabKey server. This is one example. We also introduced a more rigorous versioning system. So starting early this year, early in January, we kind of changed our numbering of our production releases. We introduced monthly releases, and we introduced versioning for every single maintenance release that we put out. And so that makes it very clear for you if you're deploying LabKey, exactly which version of LabKey do you have. And it also ensures, we made a change to ensure that no matter what build of LabKey you install, you will have at least 2 years to upgrade that build of LabKey to a more current version. So no matter what you install, you'll be able to upgrade that for a long period of time. In the way of developer improvements, we are this close to eliminating version control system from the mix. So literally next week, we will move the last bit of LabKey source code from Subversion into GitHub. This reduces complexity for anyone developing on LabKey, and it also allows everyone, no matter what you're working on, no matter which line of code you're working on, to leverage feature branch workflows, code review processes, approval workflows, many of the modern features and capabilities built into GitHub. So we're very excited about that change. We also made a change to switch from Oracle's release of OpenJDK to adopt OpenJDK. This is the community-driven, production-ready build of the Java development kit. We have standardized all of our testing, all of our development, all of our deployments on Adopt OpenJDK. The reason for this is very simple. If you want long-term support for Java, if you want security releases, maintenance updates, reliability fixes, you have to pay Oracle for those if you choose their JDK. If you choose Adopt OpenJDK, you don't. They produce those. This community-driven organization produces long-term support releases for free. So we think that's good. We don't want to make you pay for your JDK, and so we switched everything over. We encourage you to do the same with your deployments and your development. And finally, on the developer side, we've made the build much easier and much more efficient. In particular, leveraging Gradle and Artifactory, the build is now able to pull pre-built artifacts from Artifactory, and that just means you don't have to enlist in every single repository that we have. You don't have to build every single repository. You can pull down the dependencies pre-built directly from Artifactory. So let's talk about some of the new features, some of the new capabilities that we introduced over the last 12 months. Again, this is just a handful. There were hundreds of them, but these are the ones that I felt I wanted to highlight, make sure people were aware. So first of all, we improved the usability, the administration, and the capability of our authentication infrastructure. So important infrastructure piece here, allowing you to configure multiple LDAP servers if you want, multiple SAML identity identifiers, identity providers, multiple CAS servers, and do it in an easy to use, intuitive user interface. The LabKey, as you may know, allows for the creation of custom queries using our own LabKey SQL. And these queries build on other queries, reports build on queries. The ability to trace and view these dependencies is something we added over the last year. It makes it much easier to manage these queries, much easier for you to figure out that this query that I'm about to change, it has dependencies on 3 other queries and a report. So you can know what you need to test, what you need to be aware of in terms of making any changes. So LabKey is a modular-based system. All of the work that LabKey does, all of the work that our clients do with their own development gets created in a module. And then you pick and choose the modules you want to comprise your deployment. We made many improvements over the last year for this development and deployment process, the development of modules, the deployment of modules. So first off, we now allow a production deployment to update modules on the fly without shutting down the server. An administrator with the appropriate permissions can select to upgrade a module, and that gets replaced on the fly. We also, to help developers develop these modules, we have added built-in editing within LabKey server so that a development team can, for instance, use a shared instance of LabKey server to develop and test modules. So if they're developing, let's say, reports or queries, or adding JavaScript code or views or any type of resource, they can edit those directly in LabKey and use the data that's there. They can collaborate on a single server. We have within LabKey support for many different types of assays. We've had a plate designer for a long time, which has been available for neutralizing antibody assays, ELISA, ELISpot, etc. We have added plate support to what we call general assays, which are basically assays that you design yourselves. You can design them in user interface or provide them as a module resource. You can now use plates with those assays. As you heard, Rob will be talking a bit about ODBC connections and integrating LabKey with production data analysis systems that you use. This was introduced a little while ago, but in the last year, we added support for secure connections and support for hosted environments, for instance, in AWS. So just making those connections more production ready, frankly. Then the last feature I wanted to highlight is also an integration feature. So we added the ability for LabKey to act as a proxy to other web servers. This is particularly interesting for web servers that produce visualizations. So what we're seeing here is an example of a LabKey portal page showing a couple of Plotly dashboards. So 2 separate web applications are shown here. And by proxying this content, we can secure these websites and we can provide the user's credentials to those websites so that they can then securely pull data from LabKey and use that data for visualizations. So what are we going to do for the next 12 months? As usual, we don't know everything. Our clients, you, fund and ask us to add features all year long, and that will comprise a large part of the list. But we do know a few things that we want to invest our own R&D efforts into. These are, many of these we've heard from you, we've seen in the marketplace, we've seen the need, and we are putting our own R&D dollars behind them. The first, ontology integration. So this basically gives you the ability to import clinical ontologies, standard clinical ontologies, annotate individual columns with that information. This basically gives LabKey much more information about what the data actually represents. And with that information, LabKey can be smarter about harmonizing and normalizing data, presenting this information to users, creating visualizations, over time, enabling semantic search and LabKey SQL queries to be ontology aware. We also will have the ability, and this is all coming in 20.11, and will continue to be added, functionality will be added on top of that next year. The additional capability in 20.11 is a concept column, or you can think of it like an ontology lookup column, where I can, for instance, constrain all the values entered or imported into a column to a specific type of disease, to a sub-hierarchy within the ontology. Extending some of the capabilities I talked about before with module loading and module editing, we're going to integrate that, take that to the next level, integrating that with Git as well. So that in addition to editing those things, editing those resources directly within LabKey, developers will be able to save them into their distributed version control systems. LabKey is all about data management and data import, and we have many, many tools for importing data into the system, and we're going to improve that further. We're going to add more capabilities. So in the area of what we call smart diffing, is basically to improve the performance of large, in many cases, nightly uploads of data, and provide information about what has changed. Likewise, we are improving capabilities with our pipelines, integrating them with file watchers. Carl will be talking a lot about file watchers on Friday, but we're making them able to work with more pipelines. And then many of our clients are using S3 now to store a variety of different types of data. We already integrate with that very well. We want to improve that to make it integrate better with our pipelines and file watcher type triggers. And finally, my last point is just to kind of emphasize how much work we're doing in the UX department, making the LabKey server user interface more modern, more usable. We're basically taking, we're very excited about Sample Manager. We're getting great feedback about Sample Manager. We're taking everything we learned in Sample Manager and applying that to LabKey Server. We're taking capabilities of LabKey Server, moving those into Sample Manager to make it a better, stronger, and more, more effective product. All right, so with that, I'd really like to introduce my friends, Sandhya and Jason of NIAID. They are fighting the good COVID fight over there at NIH. Sandhya is the lead for clinical and laboratory informatics section at NIAID. And Jason is a contract lead at Medical Science and Computing Incorporated.
Okay, so thank you for this invitation and the kind introduction. We're really excited to present the HGRepo Informatics framework, which we developed for the NIAID Division of Intramural Research for the COVID-19 Immune Response Consortium. So this is going to be a tag team effort. And I'm going to start off with a description of the immune response research, really, because this is going to sort of inform you of how it actually affected the way how it influenced the way we deployed LabKey for HGRepo, and also the architecture, how did it influence it. So I'd like to also mention that we'll give you a brief view of the vision and summarize the presentation. Okay, so the background. The data for the COVID-19 consortium studies comes not from clinical trials, but it's really from natural history studies. And so what that means is the data is unstructured, right? Because they're coming from multiple hospital sites, both domestic and international. We have up to 14 now and growing. The data are all de-identified, however, with personal health information. There are several goals of the researchers here. Host genetics, And here we have whole genome sequencing data. We have the host response, and here you have gene expression, single cell multi-omics data, chromatin assembly, humoral signature, serology, and importantly, microbial genetics to understand what is the role of the microbiome in disease susceptibility, and how are the different strains influencing the severity. However, all of this data that's being generated has to definitely be linked back to the clinical data, the demographics, clinical profile, phenotypes, and tracking and linking and making sure that these are all then accessible to the collaborative researchers for integrative analysis. This is really key for any system that needs to be developed. So quick overview of the workflow. The consortium PIs have a memorandum of understandings with the medical institutions with whom they're partnering, receiving the samples and clinical data. They're received by over 20 PIs. So you can imagine the coordination that has to occur to distribute the samples, the clinical data to the research labs that actually perform the assays. And then they report back results to the consortium. So really the linking of all of this is so crucial and has to happen from the get-go. So these were some of what the requirements that guided us into defining and developing our scientific data management system. So there were several challenges, but also really opportunities that were part of this effort. So first of all, the researchers from the get-go designed a questionnaire, a COVID-19 patient screen. They have this sort of very well-formatted Excel document with definite sections for demographics, laboratory findings, etc., so that they would get the data, standard data in a standard format. That was their goal. However, this is a pandemic, so really not a clinical trial, right? The clinicians are more interested in treatment than they are in getting you all of the different data in exactly the format you want. And moreover, some sites have very good EMR capabilities, and others really have paper records. So really what we ended up getting is a combination of Excel formats that look nothing like the original, or even screenshots. So this is really a screenshot that had to be parsed to provide the structural data that was necessary for actually digitizing. And the data corrections came, supplemental data. And then what I want to emphasize here, we all have heard of the urgency, the COVID speed access that's necessary for the researchers to actually perform collaborative analyses. So we were sort of, really kind of like a wit's end. However, what we realized was there was really enormous opportunity here because everybody was really interested in working together. And who do I mean by everybody? Sample and clinical data curators, medical experts, data scientists and statisticians, data transformers, assay data experts, system development. Folks. So we all worked, and for sometimes we would work almost all day. We would be in contact with somebody or the other. So the system that was needed to really support this ecosystem, what I call, is something that would allow multiple data capture modes, because we wanted a sustainable system, script-based or user upload tables, or integrate with other existing sources, link patient samples to assay longitudinal data, access to diverse data sets, standardization, querying, sharing, reporting, visualizing charts and tables, the data, democratize data access for spreadsheets or APIs for programmatic access and scripting. And this is really important for enabling novel data science and distributed analyses. So really, the next generation system is what we wanted: collaborative access with role-based access, standard method of authentication, ability to publish synthetic and de-identified data sets so that we could then share the data broadly. So these were all key for us, and we looked around and we heard of LabKey, and we chose that for our clinical and sample data. We could store data in a structured, standardized, and analysis-ready data sets could be exported. But then for the assay data, since we really didn't want parsed data, but we wanted raw assay data files, we chose another data sharing platform that was outside of the NIAID firewall, easily accessible to users, and also that allowed file level sharing. But the idea is that the summary level data, assay data, is going to be brought back to LabKey for further browsing the data along with the clinical data. So I don't want to belabor this a lot here with regard to the timeline, but when we first heard of LabKey, it was in May, and we ended up deploying the enterprise version by the end of June. And then from July to now, we have this rich set of features. And really what I want to thank the LabKey, both from the acquisition personnel as well as from definitely the technical people who allowed us to implement this really creative solution that allowed us to support our COVID-19 ecosystem. So with that, I'm going to then pass off the baton to Jason, who's going to really demo, and hopefully you will see how all of this is captured in the system. So Jason?
Thank you, Sandhya. So just to recap, we had all of these different information sources and all of these different formats that were coming to us, as well as sample information. We had to transform all of that into kind of a standard structured place. And so LabKey provided that foundation onto which we're putting all of this data. And so we're just listing here on this slide some of the things that I'm going to cover in the next few minutes. And those include the fact that LabKey allowed us to define our data sets and sample properties. They allowed us to use certain common data elements like controlled vocabularies and data groupings, which I'll show you. And also we're able to build reports and views to our customer and the working group specifications. And there are cases where there's a lot of hand curating that's going on, but there are also cases where we said, if you have these two data points, you can do a calculation and infer this other data. So we have automated updates that are happening to some of our timeline data, and also data coming from the collaborative data sharing portal that Sandhya mentioned, where we have some of our assay data. So just marketing the ability to say, there's also data available in this system is possible in LabKey. And lastly, NIH authentication was a big requirement for us, and LabKey really helped us sort out what we needed to do to get that set up. And all of this is done with folder and role permission. So I'm going to share my screen now. We're going to kind of switch into a live demo mode. And what I'm going to go through with you here is we're going to start here on production. And I want to first just call attention to the fact that in addition to managing all of this data, we have just textual information, tips, and that sort of thing that we want to share with folks. And so here in LabKey Server, we're able to add wiki type information. You can see I'm linking out to a quick start guide that we host within the site. We also have some support information that lives on board LabKey and is updated frequently. And we're also able here to provide reporting on site breakdowns by site and by sample type. And people are able to use a navigator to kind of show of the data sets we have, how complete are they, or how many data points do we have in each of those. I'm just quickly showing you all of this on prod to summarize that what we're up to right now is about 12 different data sets that I'll kind of walk you through. We have about over 4,700 subjects, and the samples are over 13,000 now coming from those subjects. And so all of that is here in our kind of prescribed solution in LabKey. And I'm going to switch gears into our QA environment because I've got all of our de-identified information here so I can show it to you in screen shares. LabKey itself lends itself to doing things like, say, copy production, put it on QA, and change all the sample IDs so I can show it to others, so that we actually use those tools to create this. But let me recenter here on the clinical data. And as you've probably heard in the news, if not other places, COVID manifests itself in a number of different ways. One being how patients come into the hospital, what phenotypes they're presented with. Often, if they have a really severe case, they're being given supplemental oxygen, and there are different levels of that. And of interest to our research consortium is that information alongside demographic information, lifestyle things like smoker status, and comorbidities like did you have hypertension or diabetes or other comorbidities that you were kind of reporting in with when you started developing the COVID-19 infection. So we have divided our data sets up among those different, capturing those different criteria here on the clinical side. And then also, while the patients are being seen in the hospital, they're also collecting clinical lab data. So we have different ways of presenting that lab data as data sets here in LabKey. And lastly, we're managing our samples. We're delivering some views that I'll show you. And in particular, I'm going to show this quickly just to kind of illustrate it. And again, this is a mock-up, but we mentioned that we have another source of data where our assay data is going. And if you can imagine 20 different labs all doing kind of their own thing in terms of creating their own lab assays on these samples, they're putting them in a system where they're curating them and that sort of thing and sharing them out among the consortium. That data, once it's curated, may come back into LabKey as its own data set, but we're not there yet. And so what we're doing here is we're advertising at a patient and sample level whether there are different assays available, and then there's a link to that data. So this is kind of mocked up here, but I want to show you this is populated not by a person, but by an API with our other system. So we can have people come in here to a data set, kind of picture I'm in a regular user mode. They could click on a subject ID, and I've got one subject open here, and they see almost like a EMR all of the different data that has been collected for that patient. So I'm scanning through here, not getting yet into the details of what's in these data sets, but you can see of the ones that we've talked about, phenotypes and treatments and supplemental oxygen, I'm seeing all the data collected joined to this one patient ID. These are all the labs, so that's why it's scrolling so much. And then finally, I see the samples all collected. So this gives us a very powerful out-of-the-box method of saying, these are the different data that we're collecting. Users can go into a single patient level and kind of review everything. They can also see if assay data is available for that patient in this manner. Now, I'm going to switch gears here on you one more time and talk a little bit about as a typical LabKey user, you're going into these data sets and you have some features which I'm not going to espouse on very much, but you can create charts, you can do exports of particular interest to our group. That includes a lot of data analysts. You can export to a shell script that you can then take to something like R and make your own visualizations and connect it to other data sets. This is an important thing for us. Let me change hats on you one more time. And as we were defining all of these data sets, one of the things that we needed to do was define what goes in the data set. What is the structure, and provide some quality control on top of that. So here in our demographics data set, of importance to this particular COVID effort, we wanted to define each of these patients with whether their clinical definition was mild to severe. And you can see LabKey allowed us to put some descriptive information, so we have a shared understanding of what that clinical definition means. And also all the data that's used to kind of determine that clinical definition is here in the demographics data set alongside the typical things like age and sex. So some of the things that Adam mentioned in his updates talk was that you can use the UI not just to specify which fields are here, but important to us as we're looking at this architecture that I'm sharing with you, is we could also apply reasoning to these fields to say this is a lookup value. You can only add certain values for the COVID status, or importantly, for the most aggressive measure of supplemental oxygen. So you can see a number of these are set up not only as text or decimal, but also as controlled vocabularies through this lookup function. And that allowed us to make a listing of these controlled vocabularies. I'm showing you a sub-site where we do that in our implementation here. And I'm going to open oxygen delivery just to illustrate some of the things you can do that are quite advanced with this. So, you know, at the end of the day, we wanted to make sure that only certain values are enterable in our data set for this oxygen delivery field. And we define those here. But also, we want people to be able to analyze on that raw data or on a categorical definition of those. So if instead of getting all the way down to this granular level, we just want to say, I just want to see patients that have invasive mechanical ventilation or higher. This categorical metadata allows us to do that. So we're doing this on the list in LabKey, but if I go back to our data set, you can see that in my data set, I have the data that was entered, and then it's just simply showing me, since you entered room air, it is this category, or since you entered non-invasive, it is this category. And so this is shared out to our consortium, and they can do, this enables them to do more with the data. Similarly, I'm going to go to our phenotypes controlled vocabulary. This is something where we like using the human phenotype ontology, and this we're working very closely with LabKey to help define that ontology aware tool. This is just our sort of current approximation of that. It's not using a tool, it's just using a list like I just showed you previously. But here, our metadata, we're assigning a human phenotype ontology IDs, and we even make those linkable. So when we surface this in LabKey, our investigators can see the phenotypes that have been associated with the patient, but they can also be one click away from exploring those phenotypes. So this ability to define the data set, add definition information on a per column basis, and allow people to use that was an important part of our setup in LabKey. We use those same concepts here as we defined our samples. So we included different material types. Importantly, I want to show you that we, for many of our samples, can calculate based on a sample collection date, how many days was it from the sample symptom onset or diagnosis or admission to the hospital or ICU to that collection date. And this gives us sort of time points that are relative that can be shared among the many different sites that are contributing to this. And so this information is not really coming into us when the samples come to us, but we actually infer it from the other data sets I've shown. And we calculate it, and on a nightly basis, we actually update the samples. So as our users are perusing what samples are available, they can gain this information quite quickly. So I've shown you a few different contexts, data manager and typical user. And back in kind of that typical user role, they often have asked us for certain reports. And so our software development team has been able to deliver those. For example, I'm just showing you in summary that we are able to create a view of the sample data that I just showed you, which tracks everything at an aliquot level. But here we can roll all of those aliquots up into kind of an aggregate sample. So this is a custom view. It may not be applicable to everybody in the conference here, but this was important to us, and it was something just by using the query tools within LabKey and some of the LabKey SQL commands, we were able to build this the way our consortium wants to see it and deliver it to them through the same UI that they're using for everything else. Another similar thing, and I'll move on, is we're able to combine our data sets in a kind of pre-joined way with the samples. So here I'm showing another custom report where we take those data sets that you saw and we're joining them to sample information. And so if you can imagine, I can use out-of-the-box filter features here to build a cohort say I want to see only people over age 40 that required supplemental oxygen and had a positive outcome after that. I could apply those filters here and then say, so of those, how many do I have whole blood samples from? And basically build my cohort and then see what samples are available, all here in a single view within LabKey. I'm looking at the time, and so I think I'm going to move on here to our summary slides. Again, we've got a lot of content to cover here in these few minutes, and where we are today is, this was all stood up. None of us were what we'd consider LabKey experts back in May when this need was defined. But I would say in the months since then, our whole team has become up to speed. We've done 5 different publications out of the data that's in LabKey and being curated in LabKey, and others are on the way. We have enhancements planned, like integrating with REDCap, extending some of the dashboards I showed you, integrating with things like our server, and then of course just adding more data sets, including more genetic variant and biomarker data. Adam mentioned that there is this ongoing work with ontologies and the more robust data handling for data sets, that smart diffing that he referred to. Both of these are things that they've given us an ability to give them a voice, and we've expressed some of the things that we'd like to see as they develop this for everybody that's using LabKey, and we're quite grateful for that ability. And just bringing this to a close, some of our key takeaways for why this has been a success for our consortium. One, as I mentioned, this was stood up quickly, but allowed us to kind of progressively elaborate on the data sets. For example, as a new site came on board, we might need to add some more columns to capture the more diverse data that they're sharing with us, and we were able to progressively do that in LabKey. Another thing that contributed to the success was engaging with subject matter experts and our software developers, and really having a kind of all hands kind of approach among those roles to develop some of the features that we've seen. Prioritizing has been important because we couldn't do everything at once. Using APIs to do some of the automated work that I showed you has been important. And then finally, end user support, and even being able to deliver that support through wiki type content in LabKey was a contributor to our success. We'd like to acknowledge everyone in the Division of Intramural Research, from the stakeholders all the way to the data curators that are working on this huge effort, for engaging with us. Sandhya and I come from the Office of Cyber Infrastructure and Computational Biology, and we're thanking everyone from leadership that has helped with this to the data analysts and folks that helped with the implementation. And finally, the Advanced Biomedical Computational Science Group at National Cancer Institute that's working with that collaborative data sharing portal that I mentioned, and some of the tie-ins to LabKey. And finally, I think Sandhya mentioned it, and I've mentioned it also, the LabKey team, Adam, Hannah, many others that we can't mention, have just been really open to hearing our problems, helping us meet these challenges, and they've done a really great job for us over these past few months. So thank you, folks. I think that brings us to 1 o'clock, and we got a lot in within a few minutes.
Thank you, Jason. Thank you, Sanjay. I'd like to present Bernhard. He is going to be speaking to us about assay and study management with LabKey Server. Bernhard holds a multidisciplinary Ph.D. in biology and computer science from the University of Geneva. He has spent the last 10 years in multiple roles managing computational resources and scientific data at Nestlé. He will talk about his use of LabKey assay and study modules, as well as new functionality pertaining to data provenance tracking. Bernhard?
Thank you. Let me share my screen as well. Are you seeing my screen?
Yes, we are.
Okay, very good. So I'd like to say good evening because it's evening here, but I realize most of you it's morning, so whatever time, good day. As was said in the introduction, I'm going to talk about the use of LabKey at Nestlé and particular attention to the assay and the study modules. So I've been asked to give a few words about the institution that I work for, the company that I work for. I'm assuming that most of you have heard of Nestlé before, so I'm actually going to use this little moment to discuss the science that has been done at Nestlé over many years. So Nestlé is a company that has a rich history of innovation, starting back in the 1860s when Henri Nestlé produced one of the first specific infant foods, infant formulas, if you will. And it goes all the way to very modern things like what we released last year, which is a fully vegan cheeseburger analog that has your analog of meat, bacon, and cheese, all in a vegan manner. So all of this, of course, is founded on science and on research. And so Nestlé has a very developed R&D organization. And this R&D organization goes from basic research to produce discoveries through the development and the deployment in factories of new production mechanisms, of new products, and so on. And I myself work at Nestlé Research, which is here on the left, firmly in the discovery area. So this means that of this entire chain, we are the people who are maybe most similar to academic researchers in that we do science to a degree for science's sake, for understanding. And due to that, we're kind of a particular organization. There are about 900 people working at Nestlé Research, 50 different nationalities. We're in 4 different geographic locations. I myself am in Lausanne, Switzerland, and we produce a certain number of publications and patents and so on every year. So we tend to be a little bit like the academic people in that we want to publish as much as possible. We want to get knowledge out there in addition to what we're delivering to the company. And in so doing, we have a lot of collaborations with various partners, but also with a lot of universities. And this leads to particular way of working and a particular way of working specifically around data, the way of exchanging data that people in the academic communities are used to, a lot of ad hoc exchanges of data, a lot of different data formats, and so on. So this is the context within which we're using LabKey at Nestlé. So I'm going to talk today about 2 challenges that we are facing, that we are addressing through LabKey. The first of those challenges really is the organization of omics data as such. So to illustrate this, I'm going to take this simplified chain of events in any life science project or experiment. Generally, when we do life sciences, whether it's in a cell culture dish or in a clinical trial, we collect a sample from a living entity. So this can be harvested cells, or this can be a biopsy, or a urine sample, a blood sample from a clinical trial, and so on. There's a lot that happens in the lab on this sample. We create different aliquots, we transform these samples, extract DNA, purify proteins, and so on, in order to be able to perform measurements. And these measurements then give us some form of raw data. So typically, we have a mass spectrometer, it will give us mass spectra. We have a sequencer, it will give us raw reads. And this raw data needs to be processed in some way, and the results need to be reported in such a manner that they speak to the biological problem we're trying to address, the question we're trying to address, and are amenable to our data analytics systems and people. And so coming into this, we had a certain number of elements already in place. So we had a system that was responsible for all of the physical world tracking, basically. So we have a LIMS system. It tracks our samples, it tracks our transformations, it tracks everything that we do in order to prepare our measurements. but we didn't really have a solution for the management of the data, all of this data processing, the reporting of results, and this is where LabKey really comes in. And the complexity that we were trying to address was that it's easy to have a single system for the LIMS part because everybody collects samples, everybody does lab work on them, prepares them in some way to put them in an auto sampler or a 96 well plate or something to put them on the instrument to measure. But the data processing is a bit of a different beast because we have every technology that has its own software, its own systems, its own mechanisms to process the data and its own specificities, what type of information needs to be carried along. Much of this is file-based, much of this is vendor-specific software that cannot necessarily be automated or integrated into automatic workflows. But in the end, we really need all these results in one place where they can be collected by the data analysts and used. Additional complexity comes from the fact that, as I stated earlier, we have a very large number of collaborators, and they also send us lab data that that needs to flow into this data flow in a constructive manner so that it can be combined with the rest. As a kind of a contrast to the previous talk, we don't currently put clinical variables, all the patient information and so on into LabKey. We have other systems for that. So we're maybe a little bit upstream of what was shown earlier in that what we're looking at is people who are running analytical platforms who are collecting data from samples, processing that data and reporting the results. But in the end, we're striving for the same thing, and that is to have the results in a structured manner within the platform, within LabKey, so that it can be collected from there and either shipped to other analytical systems or directly analyzed based on LabKey itself. So how have we solved this problem in LabKey? So LabKey allows us to access files that are stored within the file root, the project level file root of a project. So what we have is basically a large file storage system which all of these platforms place their raw data on, and then if they have their own specific software, vendor software and so on to analyze this, they will process the data using their existing systems and produce results that can go into LabKey. So the solution for the file level as a first approach was simply to connect LabKey to our existing file storage system so that the files become available in LabKey and can be imported into LabKey. And then we performed a certain amount of optimizations to allow the various different platforms to easily import the data that they were already creating through import scripts. And we leverage the automatic synchronization that we have with our LIMS system to pull in all of the sample information. And this allows our platforms to basically work with generic sample identifiers. They have sample barcodes. They don't need to know that this sample comes from a subject in a clinical trial with a certain visit associated to it. They only need to know this is the identifier of their sample. And LabKey will then take care of pulling in the information it needs from our sample data. So I'm going to switch over to my browser now and give you a quick demo of what I'm talking about here. So this is our staging server. None of this data is actually live data, but it's to illustrate basically what I've just discussed. So this is a demo study, and within the demo study, we have our file storage, and this file storage, as I said, is accessible in other ways to the various platforms, and they can place their data there. And so we're going to go into the metabolomics platform here, and I don't have all the upstream part, but we have an Excel sheet in this case that has been created by this platform. And this Excel sheet is in a format that is specific to the metabolomics platform that they have been working with in the past. And we have an import script that allows us to import the metabolomics data. And this is where some of the mechanics, the technical aspects that we've put in place come into play. This is as you would see it if you have never used LabKey before. Once this is set, this becomes a default setting for that user, and they can actually just click next, and get their data in. But in the interest of illustrating what we've done here, I'm going to go in and do this configuration now and say that the participant and visit information for the data I'm uploading comes from an existing list. And so I go in here and say it's a list, and the list that I need is the sample mapping list. And this will allow LabKey to associate the barcodes and the unique identifiers of the samples in the Excel sheet that's being reported on directly into the LabKey tables. Here I'm just going to click on Finish. I'm not going to put any comments in, and as you can see, this turns into a background job that runs. And of course, there is an error because I'm doing a demo. Okay. So I'm not gonna redo this here now, but the idea is basically that we then go into the assay module, and in the assay module we have a table that contains the unique identifier of the sample that was used to upload the data. We have the participant in the clinical trial, or the sample that was the properties of the sample that are important associated with it, as well as the time points or the visit, and this all becomes available directly through the combination of the data from the samples and from the unique identifier directly in the assay, and this assay can then be uploaded into the study module in the way that you saw in the previous presentation, to be able to explore that data and combine it with data from other platforms within the same project. I'm just going to jump back to my PowerPoint and move on to the second aspect of the talk today, the second problem that we're trying to address, and this is data provenance. So we often find ourselves in a situation where we have a document with a set of results, a set of insights. This may be an internal report, this may be a paper that's been published, this may be a patent that's been submitted. But what we'd really like from a data quality point of view, from a good practice point of view, is to be able to determine where all of these results came from that are being reported here. So what did we actually use to produce this figure, and what were all the intermediate steps? And this can be relatively complex. So we have a bunch of samples that were collected. We have all our sample tracking that meets all the quality standards, but all of this element in between of obtaining raw files, processing them in a specific way, combining them together, integrating them with other data, is something we also require tracking on that was not in place prior to our use of LabKey. And this is where the new provenance module comes into play. So what we required and what we discussed with LabKey was to have the ability to record provenance between arbitrary elements within LabKey. So we have samples that are present in LabKey, we have files that are present, we have tables in the assay module, in the study module, we may have analysis reports and so on. And we'd like to have a connectivity between all of these in order to be able to trace back exactly what happened. And we'd like these connections to be annotated with the necessary information to understand what happened. So which versions of software were used, what reference databases were used, and so on. And we'd like this to be automatable in the sense that any action that is performed directly in LabKey is automatically recorded. And we'd also like to have API endpoints so that when we do have automated data processing, this automated data processing can be connected up with LabKey and can provide LabKey with the information to say that this processed file comes from this raw file, and that such and such software was used in such and such version with such and such database, and so on. So I'm going to jump back to my demo here. And I'm going to go into the proteomics data here and show, in this case, how this would be manually recorded. It's clear that the manual recording is a certain amount of overhead, and wherever possible, we would want this to happen through APIs. But the manual recording allows me to show what it is we're recording and what kind of data on provenance is then recorded within LabKey. So I'm going to take these three raw files, and I'm going to create a run. And I'm going to say these are input files. And this gives me the run builder. So the run builder is part of this provenance module. And I'm going to put in here that this is my demo run, just to give it a description. And I already have the inputs. I'm just going to add a file quickly here and say this is my output. So I now have inputs, I have files, I have outputs that are files. I could also have samples that are inputs or outputs. And I have properties here. So in the properties, I can add various properties that are important for this step that is being recorded. And I'm going to say this was an Orbitrap instrument. I'm using the human database to recognize the various peptides in my spectra. And I'm going to say this is the 2020 October version of the database. And then I click on save here, and it will save this run. And if I go to the experiment module, I will be able to see this run, this demo run here, as if it was an experiment. And if I click on it, I can view it as a graph. And I can view it in a new version of the viewer in which I have the context of these three files ran through this demo run into this, this output file. And potentially this chain can then be extended to have a large tree structure or graph structure with samples at the top, raw files, semi-processed files, processed files, and then go into the table of the assay module to show this is the origin of that tabular data, and then from there go even further and say there's an R report that was created from this, and the R report used a query over certain different tables, so that we can have the full audit report, if you will, of how a certain result was obtained. So yeah, those are the two aspects I wanted to show, to discuss and demo today. Clearly, there's still a lot of work to do. The provenance module is shaping up nicely. It's ready to complete the task we want to complete with it. The manual run builder is there. The APIs are there, but it's now in our camp here at Nestlé to continue on with the work and integrate this with all of our automated data processing solutions. The visualization of the provenance graph that I showed is still very much a work in progress, and we'll continue to develop together with LabKey the improvements on that side. Ontologies were already discussed. They're also a big subject independently of what I've shown today for us here at Nestlé. So we're very interested in seeing the evolution and the elements that were shown today in the roadmap on that front. And the study module is a module that is very much oriented towards human clinical trials or animal experiments, in that it has a participant, it has certain recording of time, whether it's continuous or visit-based, but it doesn't necessarily meet all our needs for cell-based experimentation, for recording of cell-based experiments. So we're running various options through our minds at the moment to see how we can leverage LabKey to really record that in a structured way as well, and build that data into an asset within Nestlé. So with that, I'd like to thank you all for your attention. Thank all the people at LabKey who have been instrumental in getting us to the point where we are today. And yeah, I'm ready to answer any questions you may have.
Oh, we do have a question. Question is the provenance module open to everyone?
So it is a feature in the enterprise version of LabKey, and it's still under development. So it comes with a little warning saying it's not yet a solid API, a static thing. It's still going through frequent modifications, but it's available for use in the enterprise version.
Great.
Yeah, and this is Adam. I'll just add that our plan is to make that available to all of our enterprise clients in 2021, once we've kind of stabilized it and established those APIs.
All right, so I'm Rob Morphew. I'm a technical program manager here at LabKey, and I'm going to be talking to you about reporting and visualizations via ODBC. So we will cover a brief introduction about what ODBC is and why it's important, what we've done new involving ODBC. We'll kind of broaden the spectrum a little bit and talk about our supported integrations, some beyond ODBC, and then kind of go into some screenshots of our new administration interfaces and a demo. So external tools integrations. They allow us to create very complex and customized reports and visualizations outside of LabKey. We employ well-established products with deep knowledge bases. We utilize live data in these visualizations. Integrate with existing reporting infrastructure. So we know that a lot of people actually use products like Excel and Tableau and other products to visualize this information. There's the convenience factor. You don't always have to export and import into these tools. You can actually view live data right in the tool that you're using. All permissions are respected. So if you've spent a lot of time defining your permission models within LabKey, those permissions are respected within the relevant external application. Same with PHI and auditing. And now we have, and we'll get to this in a little bit, we have cloud-enabled options. So what is ODBC? ODBC stands for Open Database Connectivity. It allows you to integrate with applications like Excel and Tableau, Microsoft products, and the like. It's been around for quite some time. LabKey API, which stands for Application Programming Interface. This allows you to integrate with RStudio, and as Adam had mentioned in his slides, Plotly Dash through Python. And also we offer the LabKey JDBC driver, which at the moment allows us to integrate with Spotfire. So what's new? We have enabled cloud access. So we've spent a lot of time on security, and we continue to do that this year. So part of that option was actually enabling ODBC, secure ODBC access to the cloud. There was extra steps, if you will, to allow hosted LabKey instances access via ODBC. So we spent a lot of time on that. We enhanced our secure ODBC admin interfaces to be a little more helpful and to accommodate the new interfaces we needed for cloud. And then we tested Stata. We continue to test Stata to make sure that this new tool, it's not a necessary new tool, but this new tool, ODBC access, will be handled very well in LabKey. So let's talk briefly about our supported integrations. Obviously Excel. This uses our ODBC integration, respects the permissions models like all of our integrations. It allows users to build reports with live LabKey data. So some of you who are, have done this before with other databases, you're able to essentially refresh that data, and it's live data that gets pulled in. It's not an export. There are ways of using LabKey SQL directly in reports. Being in Excel, someone who likes Excel, you're able to use pivot tables, pivot charts, and all the other tools, formulas that you're used to using. Tableau also uses our ODBC integration. It lists us as a database within its visualization. Also lists all of our schemas and queries so that you can have access to that and the report building. And then uses standard Tableau chart creation options, measures over dimensions. There are some additional ODBC clients that we've confirmed. Microsoft Access, MATLAB, SQL Server reporting services, and Stata. We also here reported compatibility with JUMP, VBScript, which I've done, Primal SQL, and SQL DBX. As I said, we're going to talk a little bit briefly about the other integrations. So one integration that we have is with RStudio through our LabKey API. Provides a richer sandbox R development environment. You can install your own private packages. Again, this respects your permissions that you've set up in LabKey server, and works with the open source or commercial RStudio licenses. You can interact with Shiny, which allows you to build visualizations as web pages. You can interact with TIBCO Spotfire via our JDBC driver. Again, this builds visualizations directly with LabKey server and with live data, providing a connection URL, username, and password. So, ODBC administration. How do we interact with ODBC? LabKey didn't provide a driver per se like we did with JDBC. What we did was we integrated with PostgreSQL's wire protocol, which allowed us to expose the LabKey virtual schema this way. So ODBC client applications will execute SQL against the schema through this interface. So if you're going to use ODBC with your LabKey system, you'll actually be using the PostgreSQL ODBC driver. We've expanded our admin and advanced security options within LabKey server, predominantly to make sure that we offer secure access to the cloud. This is can be accomplished with piggybacking on Tomcat's HTTP certificate, but more often than not requires a self-signed certificate that is separate and distinct to allow access to the cloud environment. We also thought it was kind of necessary to put some more tools in for users to give them hints on how to actually connect via an ODBC DSN. So we provided a menu for users that are authorized under the external tools access menu, and this will provide the credentials that you will need to connect to LabKey via ODBC. All right, so how do we get into LabKey via ODBC? For some of you, this might be review. You're able to open up ODBC, define what's called a DSN through the ODBC Data Source Administrator, and then from there, gain access to a project or folder within LabKey via the database server username credentials. Okay, so I think we'll go ahead and start with ODBC and Tableau first. I'm going to go ahead and demo both live systems with my local instance of LabKey. What we're going to do is we're going to try to produce this visualization, which is showing you CD4 levels with HIV positive and HIV negative participants over time. So you can see the visit day at the bottom. So let me just stop sharing really quick here. And bring up Tableau and share the screen again. There we go. Some of you who are familiar with Tableau know how to connect via to a server menu. The way you'll connect to LabKey is through an ODBC DSN. On this machine, I've actually already defined an ODBC data source under my system DSN called LabKey Study. So what this does is it allows us to connect to a folder on LabKey server using the appropriate credentials. Of course, if you're connecting via TLS, you will use the SSL mode options to connect securely. My particular instance isn't running SSL at the moment. But you will need a DSN to connect to each folder that you want to gain access to. Make sure my connection is successful. All right, so through Tableau, we'll go to other databases, ODBC. That's actually within the larger menuing option. This is just remembered that I'm actually interested in ODBC. You'll select your DSN from the list. Click connect. And in this case, it's remembered my connection details from the DSN. So let's sign in. All right, and then this gives you a kind of a blank canvas to Tableau. So here we're going to search for our schema. And in this case, I'm going to choose study. And then I'm going to search for a query that I prepared for this demo called, let's see. There we go. Physical exam demographics. So you can click the update now. And notice that this is actually live data. You can actually have an extract that Tableau will save to your local machine, but in this case, we're going to use live. Then we'll select a workbook. And you can see that on the left-hand side, Tableau has already categorized my measures and dimensions from the columns that were in my LabKey query. And in this case, we are going to, got little notes here. We are going to drag CD4 to rows. And in this case, I'm interested in it being a dimension. And then visit day will be our time element. And you can see that Tableau already has inferred this as a scatter plot for me, which is great. And then there's a really nice function here in Tableau where you can actually color these little dots without doing any extra work by dragging a separate column over this marks area. So I'm going to drag it over color. And you can see that Tableau has already designated the HIV negative and HIV positive bubbles in my scatter plot. Let's see, let's add some trend lines. And of course, so I have this for later. I'm going to just give it a name. All right. So there you are. This is live data from LabKey server that can be refreshed within Tableau. So let's try to do this in Excel. Excel was a little bit more difficult, but still doable, especially for the Excel geeks out there. In Excel, there's a similar way of getting data into a worksheet. You want to go to the data ribbon, go to get data, and go to other data sources from ODBC. All right, and then we'll select our DSN called LabKey Study. And similar to Tableau, we will have a tree control of all of the elements in our study folder. So we're going to pick study, navigate down to the query that I want. You can see that it previews the data for you. And then it's going to load the data into cell A. All right. Great. So as I mentioned, in Excel, we have to do a little extra work to produce the same viz, as they call it in Tableau. I know there's probably a million other ways to do this. I actually attempted to do it through Power Query Editor and pivot tables and the like, but I found it to be a lot easier just to create two separate columns. Now, you'll notice this is, again, live data, so we can just put our cursor anywhere in the table, click refresh, and down here you can see it's running a background query where it's pulling the data to update our spreadsheet here. That refresh, as some of you might know, can be actually linked to opening up an Excel workbook, and it'll refresh all of the linked data when you open it. Or you can have it on some sort of manual button push, or even on a time. So let's create those two columns that I need. To do this, we're going to need some formulas. And I'm not going to type them out. I'm just going to cut and paste them in here. There we go. Great. So given those two columns, we can create a similar scatter plot. So I'm going to select only 3 columns for this. I often grabbed all the columns and realized I can't do what I want to do. We're going to insert a scatter plot. And voila, Excel does something similar, although the colors are inverted. So you can see that it's already identified blue dots as HIV positive and orange dots as HIV negative. Let's grab that title that we want to put in here. All right, and then I think I'm missing, yes I am, I'm missing my trend lines. So let's add some trend lines. Actually, I found that if you select the blue there, you'll get a trend line that you can barely see. And select these little orange guys, and you can add that trend line. And there you have it. Excel integration with LabKey through our ODBC integration. Let's see, let me just shrink that down and bring up my presentation again. All right. So that's it for the demo. Any questions?
So we do have 3 or 4 questions. This is Adam again. So I think Rob and I will kind of tag team on the answers here. If you have a question and you've been holding back, go ahead and fill it in the Q&A section. But just going through the ones we've gotten already. Does the ODBC connection work if you have Microsoft SQL Server for your database? And the simple answer there is yes, absolutely. So the underlying database really that you're using has nothing to do with the fact that we happen to be using the Postgres protocol effectively in the front door. So we're only using that protocol to get the queries and the commands from the application to LabKey, kind of in place of what would normally be an HTTP request. So instead, we're just using a socket with that protocol. We implemented that protocol. It's not going to the underlying database in any way, shape, or form. So you can use this mechanism to access SQL Server data, MySQL, Oracle, Redshift, Postgres, any of the queries that would run on any underlying database. So another question is, can we use ODBC data sources to bring non-LabKey or external ODBC data sources, data source queries into LabKey, such as a LabKey report or as a data grid. And that one is basically no. So the ODBC connection is really used just to get data from LabKey into external data analysis tools, the ones that, like the ones that Robert has been showing. However, LabKey uses JDBC to provide access to a wide variety of external data sources. So as I kind of just mentioned, if you have data sources, databases, relational databases in the enterprise that are Postgres, SQL Server, SAS Share, Oracle, MySQL, or Redshift, any of those data sources can be configured within LabKey so that data grids and reports and our APIs and the schema browser and custom queries can all access data in any of those databases. And so I encourage you to look at, just go to labkey.org and search our public documentation for external schemas and external data sources, and that will provide a lot of information there. Another question is, how much impact does working on a live server have on other users? So I don't know, Robert, you want to take that, or I can?
Both. Well, I mean, we've done some amount of testing. The impact doesn't seem to be great. Adam, I don't know if you've run in any situation where you've created some vizzes or Excel queries against LabKey that have put an impact on there that we're not normally used to seeing.
No, it's really, there's nothing that special about this. There's not a lot of overhead basically in that ODBC protocol. So it's going to be very similar to an export or using one of our APIs like select rows to pull down data, or using JDBC or using any of these mechanisms. You're essentially doing a similar amount of work. Now there is there's a difference between, say, going to a LabKey grid, which typically those grids are going to be paged in the sense that, we only show the first 100 rows, let's say, when you first get to a query. Whereas with ODBC and exports and JDBC and the APIs, oftentimes we're sending all the data. But really the bottleneck is, as usual, is going to be on that query, whatever query you've implemented. If it's a long-running query, if it's a complex query, then it may take a little while. Obviously, if you have 10 million rows, it's going to take a lot longer than if you had just sent back 100 rows or if you have a smaller data set. But there's really no inherent overhead to speak of, or unusual overhead with this protocol. In fact, it's probably more compact. Sending the results back is probably more compact than, say, the JSON results that typically we send back via our APIs. Let's see. And again, if there are any other questions, please feel free to type them. We've got a question. Can I share the Excel file? Can I share the Excel files with coworkers so they can use them?
Yes, you can. Yes, you can. You can actually share the Excel files and the Tableau workbooks with coworkers. Of course, the DSN that is set up on the local machine is going to be the DSN that is used and respected as far as permissions are concerned. So if you shared the Excel workbook with a colleague, your credentials would not go with that, and we would require the DSN that is set up on that local machine. Of course, the same DSN has to be set up against this, essentially the same parameters.
All right, if there are no other questions, then I think, Rob, you're going to close it out?
Yes, I am. So I think what I'm going to do is I'm going to let you know what we're doing tomorrow. So tomorrow we're going to open the day with LabKey Biologics update from Bernie. And then we're going to talk about ELN, and Bernie's going to walk us through that. Then at 10 o'clock, we're going to have the quest for an ELN that we actually want to use, presented by Tara at Just. And then at 10:30, introduction to Panorama and LabKey support for targeted mass spectrometry by Josh. So please join us tomorrow at 9 a.m. for day 2 of our LabKey user conference.
Thank you, everybody. Thank you, Rob.
Bye.
Thank you to all the speakers today.